Yeah. We should be ready to go. You have more people speak other than they're dropped.
Nice to meet you.
Thanks for having me.
Thanks for coming down. Erica organized the whole thing.
Well, and I did the easy part, so. Actually, I was gonna ask, do you prefer Greg Glass and do you prefer them Rosa in Spanish or like?
Just do the business.
Right.
Well, unless you speak Spanish as your first language.
I would say. Roll at least is an easy.
Yeah. Usually I wait five. Usually we'll wait
Oh,
five.
yeah, you're 5 minutes. We're 10 minutes Berkeley time, same.
I forgot when I was doing Ellen's contact in the Zoom room and I was like, there's no one here. Hey, you know, parents say, Sadie, which is, yeah, I'm not.
Good, how are you doing?
So did you come down yesterday or did you come down this week?
Last night, yeah. Yeah, it was nice to relax, yeah.
Did you guys get together yesterday?
No, my flight ended up getting canceled, pushed back later, like it was the week of cancellation.
What else got canceled? What was it?
One of the EPA stars.
Oh,
Kind of star. What's gonna happen? So yeah, we're just,
So it was an EPA thing.
we had realized.
Can they do that? in the middle? Or
They're trying to sell, they're trying to feel that.
was it in the middle of the year or was it at a?
It was towards the end, it was like the third year of the. So you.
And in mid-year?
Yeah, they just stopped for, but they had done it originally, Rosh's grant, not the week before, so we guys had sense. Yeah, I have, I think it's gonna be appealed, you know, process when I go to class. But. Yeah.
Does the university have a legal case?
So Berkeley had clear to all right now, they're gonna need to put out.
And within a year, it's like a contract. It's a grant.
Yes.
Should be.
Are you finished your day, Matt?
The universities make.
I'd like to give everybody an extension.
Yeah, that's the-
Hired.
We had hired someone Friday.
Are you done?
You're not done?
So yeah, we're not keeping.
I have to send it next Friday,
Okay.
and then, but then the email.
So my flight was pretty, you know, just
Yeah.
changed it.
Oh, now I'm like, looking
Well, no,
They gave me a later one that actually got on, I was like, well, that's too late. I have to give that. You know?
Still flying to Burbank or do you go to LAX?
Down to Burbank.
yeah.
house.
Yeah.
Yeah.
Yeah, and I flew to Oakland too. So they're doing some. Yeah.
Some of those. I kept telling.
So was it the house if you were like?
So
No, it was Sunday.
good morning. Welcome to the Southern California Environmental Health Sciences Center seminar for this month. This will be our last seminar of the year, and we've got a special treat. Rosemarie de la Rosa is going to speak, and Erica is going to introduce her. So take it away.
Great. And just to clarify, Rob, it's the last seminar speaker of the academic year, right? Not the calendar.
Yes,
So stay tuned for the fall.
unless you have something really important. So.
Let's hope not. So it is my pleasure to introduce Dr. Rosemarie de la Rosa, who is an assistant professor in the Division of Environmental Health Sciences at UC Berkeley, where she also got her PhD and MPH in EHS. And we actually were there at the same time getting our degrees. She got her bachelor's degree in biology from MIT. Her primary research goal is to understand how social environment and context during childhood gets under the skin and influences susceptibility to toxic effects of environmental pollutants over the life course. She's a laboratory scientist whose research lies at the intersection of environmental toxicology, epidemiology, and molecular biology. Health equity and environmental justice are at the center of her research program. Current research projects include collaborations with investigators at UCSF, examining the relationship between adverse childhood experiences, or ACEs, resilience, and biomarkers of toxic stress using allostatic load framework. She also has projects evaluating the joint effects of psychological stress and air pollution on health outcomes, and stress-related biomarkers in children, adolescents, and caregivers from diverse communities. So give Dr. De La Rosa a warm welcome.
Hi, everyone. I'd like to thank Dr. Garcia and also the Center for inviting me to speak today. And thank you all for joining and listening to what I've been working on for the last, decade of my career. So as my talk title suggests, I'm going to be talking about the biological burden of both chemical and non-chemical stressors in pediatric populations. So more of my molecular epi training. But I want to kind of just bring you to understand why I'm interested in this topic. So for those of you I met this morning, I grew up in New York City in the Bronx, and that is a borough within New York in case you're not familiar with it. And it is a high prevalence of Black and Latino population. It's predominantly 80% Black and Latino. And unfortunately, when I was growing up, it had the highest asthma prevalence, county in the country. And so the question when I was growing up was, why are my neighbors, my friends, having high prevalence of asthma, right, in the community? And as you start to reflect on what is going on in this environment, this was the Cross Bronx Expressway. I've been fighting with this laser all this time. I'm just gonna point out, I don't think I need a pointer to show there's a freeway cutting through what you'd see people living right next to it. And I drove on this every day to go to school. There's not a lot of green space here. And so this is a product of the Moses type eminent domain, where they pretty much put freeways throughout 3 intersecting freeways with a lot of trucks. And so it's not surprising that there is a high asthma prevalence because of the built environment that's there. Now, at the same time, I remember while I was in graduate school, a faculty member asking, what makes you so special? Why are you sitting in front of me and not, someone else? And so that made me start to think about, well, what were some things that were present in my community that were also buffers? And so I just, throughout my time, I wanted to think about not only the environmental hazards that might be present, but what are some of the buffers that we could think about that are helping to mitigate some of the effects of, these obvious exposures like air pollution. So as Dr. Garcia already introduced, I have a research program that is a laboratory-based program at UC Berkeley that really couples not only environmental toxicology, but also leveraging, so toxicology, what I mean is like experimental models in cells, and then also coupling that with some human population studies, and also thinking about social determinants of health to think about how all of those intersect to then influence disease susceptibility. And for those of you who like, conceptual model, the models that I really think about in my group are the social ecological model. So thinking about how policies can influence, exposure at the neighborhood level and then trickle down to the family context and then variables at the individual. And also that together with life course, right? So we know that that is always dynamic throughout, the trajectory of one's individual life course, and that can influence someone's susceptibility to disease. I tend to look at asthma, and I will describe why that is. But I think we know that there's also the issue of not only just cumulative over time, but the question is, how is that happening, right? How are these things occurring? And so this concept of biological embedding of these exposures is another area that I've tried to really focus in, given my biology training. But that's really hard to capture. And so I'm hoping that through this talk, I share that journey of how I'm trying to understand that biological embedding process. We also know that oftentimes we're looking at one exposure at a time, but that's not necessarily realistic of the human experience, right? We're simultaneously exposed to a lot of different factors. And we know that, but the problem is understanding the impacts and the risk is really hard. And so I think the EPA is trying to understand these cumulative impacts, or what do I mean by cumulative impacts? It's that combination of chemical and non-chemical, and I'll describe some examples of what I mean by non-chemical later on, to think about how that is impacting human health. And this is really important within an environmental justice context, and that's actually why that came to be, because environmental justice communities were really arguing that it's always the overburdened communities that are really facing, multiple simultaneous exposures. So thinking about this risk framework at one exposure at a time is not really the experiences that they're having. And I think to just provide an example, this is evidence from the Children's Health Study in Southern California. I don't think I need to explain the study to this group, but just some of the early examples that psychosocial stress, which is the exposure that I look at as well, thinking about how that might modify the effects of an air pollutant. So in this case, with traffic related to air pollution. And so on the x-axis here, you have a parental stress quartile. So that is on the X, and then on the Y, you're looking at the hazard ratio of thinking about among kids. So it was over 2,500 kids without asthma. And so you're trying to follow over time the incident cases and that association with traffic related air pollution. As the quartile of parental stress reported increases, you're seeing that that relationship is increasing as well, suggesting some modification of the effects of air pollution by parental stress. So this is just to demonstrate the example that we know that this exists. There's more studies that have been emerging over time. But the question is how do you measure that? What's that process? And so I just wanted to put forth a framework, or share a framework by Dr. Rachel Frosch, to think about how that might happen in riskscapes or communities that are exposed simultaneously both to environmental pollutants and psychosocial or non-chemical stressors. So to begin with introducing that framework, I just wanted to start by sharing the exposure health response pathway that I think most people are familiar with here, where you would have your exposure that happens because some pollutant source, has emitted some exposure, and over time, there's an internal dose that becomes internalized, and then that elicits some response. And then we eventually see some health effects that occur. Now, that exposure needs to meet a certain threshold, right, to elicit a health response. And the question there is, well, are there factors that may change that, susceptibility or that response to a pollutant exposure? And that's where these factors at the community level and at the individual level are hypothesized to potentially have some increased chronic stress, and then potentially increased allostatic load, which I will describe what that concept is, which then modifies one's susceptibility to pollutant exposures. And is that increase in susceptibility to pollutant exposures that may then increase one's ability to increase the risk of health outcomes. But that's a interesting framework. How do you measure that, right? And so from my perspective, I've been really focused at trying to understand what is that individual allostatic load, and what are these health effects and responses? Can you measure that to then think about what is that cumulative impact of chemical and non-chemical stressors? Early on in my career as a graduate student, I thought I could do this in a dish. And so I thought, well, what is the stress response, right? So that is, or that how are psychosocial stressors even embodied? And so you're thinking, well, maybe that's a stressor, that then you have to think about what is the mechanism by which stress, acts. And so when you experience a stressful experience, there's a fight or flight response that triggers secretion of this hormone cortisol by the HPA axis, to the hypothalamic pituitary axis. And so thinking about this hormone, sorry about the bubble, ends up then secreting cortisol, and then it'll find this receptor in its cell. And you have this receptor in almost every cell, and that's the glucocorticoid receptor. And so thinking about, well, do chemicals actually act on the same receptor was an idea that I wanted to kind of explore, to think about, well, that's one commonality where you may have some intersection. So I, created a cell line that essentially is like, think about a firefly, right? So it like lights up, and so you can use that, harness that gene that creates like a light, and you can put the response element before it that is responsive to activation by the receptor of cortisol, and see if things turn on and off. And so, for example, if you have cortisol within the system and you treat your cells, you should see some signal that turns on. If you have some chemical that mimics cortisol, you should also see something that activates the system. And then if you have something that blocks that response of cortisol, you should see no light, no signal. And so this is just a tool that we use sometimes to identify chemicals that are acting or inhibiting a response of some gene or receptor that we're looking for. And so I wanted to then see if, well, do chemicals do this? And at the time when I treated with 176 structurally diverse environmental compounds, I actually didn't see anything that activated or inhibited the response of the receptor. But what was unique was that when I had the presence of cortisol and chemicals in the same dish, I did see that these three chemicals activated the receptor beyond what cortisol could do, suggesting that there's a good potentiation effect. Why do we care? Well, one of these no longer is in products, but it was triclocarbon. So that's like an antimicrobial agent. And so those are things that I think, could suggest that there's chemicals that may also act on similar pathways as our stress pathways. And so for a bit, we were calling these compounds stressogens to correlate it to like obesogens, thinking about these chemicals are acting on similar pathways. But that's in a dish, and I think that's important, with the these non-animal models, so NAMs coming out and thinking about this, and I'm still collaborating with colleagues to think about how do you maybe reflect more like a human-like scenario. But the reality is I'm really interested in understanding how to do this at the population level. But that's also challenging. So I'm just gonna provide an example as to how I'm thinking about this and moving forward to think about these intersections. But first I want to introduce like this double hit hypothesis to kind of reinforce the idea. So if you have, and I would highly suggest looking at this review if you want more details by Julia Frosky at Northeastern. So you have a population at baseline, and what these red individuals are individuals, if you're measuring, for example, biomarker or some clinically relevant outcome. This may be the baseline risk in a population. So let's say you're measuring blood pressure. And so then this is maybe the prevalence in the general population. Sorry, I don't know what happened. But then let's say there are some baseline intrinsic factors that may increase the risk. For example, as you age, you may end up increasing the number of individuals who have, elevated blood pressure. Now let's think about, are there extrinsic factors that can then also act upon this to increase the risk? So for example, if you're also experiencing stress daily, then does that shift the population even more, right? So that's the question of, what are these extrinsic factors that may be kind of shifting individuals to have physiologic parameters that are higher and also at greater risk. But the question is then, what biomarkers are you going to use? And so I think that's where I encourage everyone to think about that mechanism, their causal framework, and what kind of disease outcomes they're looking for to see where that might intersect. Now, for me, I'm thinking about stress, and I've kind of explained that there's an increase in cortisol in response to stress. And so there are some downstream measures that are a little bit more stable than cortisol to measure, and I'll discuss what those are. And so that's where this allostatic load concept comes in, and I'll discuss that a bit more. As well as you may have seen in the literature, things like cellular aging processes related to telomere length or genetic age acceleration. Today, I'm going to just highlight a project on telomere length at the end of my talk. And then also thinking about conceptually, is there evidence that air pollution also acts on these pathways? I would say, and I'll show you that there is evidence definitely of air pollution acting on some of these cellular aging pathways, but less has been done with allostatic load. And so I'm going to show you one, how these things are independently maybe associated with allostatic load. And then at the end, I'll show you how there may be actually effect modification happening with these two variables. So to begin, what is allostatic load? And so this was a framework that came from Bruce McEwen, thinking about chronic wear and tear on the system. So we often demonize stress like this is a bad thing, but actually it is something that was evolutionarily necessary for you to kind of run away. If there's something scary or, or hazardous in your environment, you need to mobilize and move, right? However, when it doesn't shut off, that is when you have an issue, right? Where you kind of have this chronic activation and secretion of these hormones, cortisol and epinephrine. And so your body is always kind of trying to maintain homeostasis. And so as you can see, there's some stressful stimuli that one, you have some activation and it'll, eventually go back. But there are some that elicit different responses, right? So like not every stressor is the same. So I don't want to trivialize anyone's trauma, but for example, taking a test may be different than, having something present every day in your life, like not having the funds or needs met that you need for survival. And as you can see, if this consistently happens, what ends up happening is you erode at the biology or the physiology that is in place. And because of this erosion and new set points that emerge, you end up having new set points of different baseline levels of these physiologic parameters. And so theoretically, that difference between the new set point and your baseline, your theoretical baseline, is considered allostatic load. Now, this is a concept. It's a lot harder to actually measure in reality. And so people try to use different tools. Some individuals will try to measure these primary outcomes or these the cortisol, epinephrine, some of these hormones that are kind of the initial response. But there's challenges that I can go into. Cortisol, let's just think about as secreted in a diurnal cycle. So imagine you have to capture someone, when they wake up 30 minutes later and then throughout the day and probably 3 times and, 3 times to get a valid assessment. And I think in a pediatric population and also in clinical practice, that seems pretty challenging. We're working on some implementing measures of hair, and so I'm happy to chat more about it. But as you can imagine, there's challenges in general with assessing those primary mediators. So oftentimes in the field, you'll see measurements of secondary outcomes, or they're capturing dysregulation across, other physiologic systems like cardiovascular, metabolic, and immune. And usually you try to capture multiple biomarkers across the domains to get a sense of better dysregulation across them. Now that I will note, the reason we care is because allostatic load has been in adults has been associated with morbidity, mortality, and this tool though is really difficult to measure in kids because they're always changing, right? Their biology is always like not static, and so getting biomarkers and operationalizing that is a challenge of the field. I think we need to kind of think more about it. But I don't think that we should not explore how to do this. And so for when I was beginning this work, I thought, well, what kind of non-chemical stressors should I think about? As you can see now, there's a variety of different exposures that people have looked at, but in particular, I was interested in looking at adverse childhood experiences. One, because I had access to a really unique data set or a cohort I still work with. And also there's pretty robust literature on the association between adverse childhood experiences and health outcomes throughout the life course. What do I mean by adverse childhood experiences? So adverse childhood experiences capture traumatic life events that occur between the ages of 0 and 18 across 3 domains, mainly abuse, neglect, and household challenges. And you can see which of these are captured here. But this was validated mostly for adults. And so I'll touch on tools that we're using that have been validated for kids. But the reason we care again is because the landmark Kaiser study found that ACEs having 4 or more of these, reporting them, was associated with, at the time, 7 of the 10 leading causes of death. But this is an updated table based on ACEs Aware, which is an initiative across the state, which we can talk about with anyone who's interested, has updated this to more recent data. So this is telling you that ACEs is important, right? But do we have to wait until adulthood to start establishing a relationship, to understand what is going on and identifying kids who may be more at risk for developing these adverse health outcomes later in life? So that was a question that was posited by the team at UCSF and Center for Youth Wellness when we launched the study of the Pediatric ACEs and Resiliency Study, which is a cohort that was launched out of a primary care clinic. So it's a federally qualified health center. It started with 555 kids with a lot of, we've screened them, and there's as you can see, we've been following them for 5 years. We just finished the 5-year recruitment. And we have a lot of questionnaire data at the baseline, as well as clinical exams, biomarkers, and then there's some interesting intervention work that I can talk about at another time. But as you can see, there's the key parts being that we've been following them for quite a while. So we can then start to think about trajectories and biomarkers that we've captured and what those relationships are. Also, some key points to note, it's a high burden population. So about 76% of the children have at least one of the adversities that I mentioned before. And as you can see, we do most of our work in populations that are not white. So we are trying to understand, how do you identify mitigation factors within these overburdened communities? So prior to my postdoc, one of the initial studies had looked at this tool, which is the Pearl Screener. It's actually being implemented across the state now because of this study, in which they assess the original 10 items that I had shared, plus these 7 related life events, which are a more holistic view to kind of capture a toxic stress. We're trying to move away from that term, but toxic stress events that are occurring. And so, as you can imagine, you can have the original ACEs, or 0 to 17 items are included in this Pearl Screener. And so this original study by Tucker et al. Looked at the relationship between Pearl scores and many of these health outcomes and found an association. But the question is, and at baseline, the question is how, and are we able to identify any biomarkers that may be related to this Pearl score and also adverse health outcomes? Because that's a limitation in the field where we haven't really looked at the exposure and the outcome relationships with many of these stress-related biomarkers. So I conducted a study that tried to operationalize allostatic load in children. There's no standard, as you can tell. And even myself, I am guilty of taking biomarkers that I have available and conceptualizing how they would fall across the 4 domains. And so with the goal of identifying some dysregulation across immune, metabolic, and cardiovascular outcomes. Many of these are empirical from plasma levels that we've measured in the lab, that from the plasma that was collected from the kids. with a few such as the systolic blood pressure and like resting heart rate were based on clinical assessment. We also have other clinical labs, but as many people who work on pediatric populations know, you can only work or collect some of these things, the amount of blood you need for these assessments after a certain age. So that's only in kids who are above the age of 5, whereas the range of the kids that we were working with were 0 to 11 years old. So some of the younger kids, we couldn't get all of these clinical measures, but in this study, we limited to these biomarkers because we did have it on all of our kids that were at baseline. So the goals of the study, and I'm not going to get into it much, was to assess allostatic load in children. What we learned was biomarkers change quite a bit over time, and so that is something to keep in mind. That it's actually not a linear relationship with age. So if you're trying to operationalize allostatic load in kids, it's a challenge, and just be mindful of that. Second, I wanted to look at how ACE exposure related to allostatic load, and then third, I wanted to look at how this allostatic load might relate to some of the outcomes that we had in the study. And just to summarize, it ended up being a study of 207 kids. And for anyone who's interested in understanding how we operationalize allostatic load, we define high risk categories for all the biomarkers that were included, and denoted whether if you were in a high risk category. So for example, you would end up with a score of 1, and then the number of biomarkers that were denoted as risk, you'd sum across and end up with a score ranging from 0 to 12. And then this was our distribution. On average, it looks like kids have around 2 of these biomarkers that are at risk, which is surprising. They're kids, right? So it's denoting that we're already seeing indication. And I remember you were saying, well, your kids can't have hypertension, and that was actually not true. A number of kids actually had the thresholds for hypertension. So it's just concerning. And so we want to obviously think about what's driving that high burden so we can eventually reduce it. Also, like I noted, there's strange dynamics with age that I can discuss, but it is non-linear. And then we also saw relationships with caregiver education, where having the lowest category of education was related to having the highest allostatic load. And that's not too surprising. It tells us it's kind of working as expected, because social economic status is probably the strongest indicator or association in the literature with allostatic load. When we started looking at the Pearl score or ACEs, we saw that there were statistically significant associations with these exposures and allostatic load, with it being higher, allostatic load being higher. But what was also a bit interesting, and we need to dig into a little bit more, is that it looked like the categories just above 0 had slightly higher allostatic load compared to children who had more adversity. And that has actually been seen in other studies and literature, potentially because I think, again, we need to think more about it, but there can be some ceiling effect, in which essentially the first event may be a bit more shocking. And then as you kind of learn to cope, you may have, different responses that are a little bit more attenuated. There's also a lot of limitations with this study where we don't know when the event occurred. So there's a lot more that we need to unpack with this.
I had a question related to that. I was wondering about the timing of the ACEs versus the timing of the assessments of the plasma.
Yeah.
And all the biomarkers in the plasma that you're using. When you made the cutoffs for each of the biomarkers, was it in maybe a subpopulation that hadn't had any ACEs or anything like that, or you were looking at the whole population?
Yeah, so when we did the cutoffs, we did everyone. So it is an interesting point where you could look at what is normal, right? And that's a challenge. We don't have norms. So I think the field is also working on that actively right now to just figure out what those norms are, to then see how, and we also don't have timing from event, which is a big limitation in general of PROS or ACEs screening broadly. We don't know when the event happened, right? Did it just happen yesterday? Did it happen years ago, right? It's just a yes, no. And it's intentional because it's in the primary care setting to be brief and fast so we can get a score, but it doesn't give us any of this information that as epidemiologists would want to know when this happened, so we can adjust all these things. But yeah, it is a limitation of the screener. Thanks. We also tried to look at individual items that may be related to this variable, and we did see indication that reporting caregiver mental illness in the home was related to higher allostatic load. We still that there's more to be done there as well, but it highlights the role of caregiving, and so we do have caregiving intervention studies that we can then follow up on and see what part of that caregiving relationship may be at play here. But that's just to showcase that individual items that may have some relationship. And then lastly, I was interested in understanding, I know it's a cross-sectional study, but I just wanted to see if there were any indication of relationships with this measure and any of the health outcomes that we had in the study. We had the parent proxy of promise for the children, and we saw that having higher allostatic load was associated with lower promise, and so lower promise is actually a negative thing, and so that's just a global health measure. So that generally says that having a higher allostatic load is not good for your health. And then we also did see an association with having higher allostatic load was associated with increased odds of obesity. That could be because of like the high metabolic profile of biomarkers included. But I think in order obviously to kind of understand that more causally, we would need to follow kids over time that didn't have disease and see how this biomarker trends with it as well. But all that to say that the relationship between child allostatic load and age may be nonlinear between these ages, and children with 1 to 2 had the higher allostatic load compared to a score of 0. We think that caregiver mental illness may be something to look into a little bit further and its relationship with allostatic load. And we did see some health outcomes that were associated with this measure. It was the first study to simultaneously look at the relationships between all of these three in the same study. But as I said, since we are following these kids over time, we should kind of tease these relationships apart in a longitudinal study. But then I also told you that I was going to think about not only, I think we understand that psychosocial stress may act on these, may increase allostatic load, but less work has been done to look at the relationship between air pollution and allostatic load. That's not to say that there's no studies. There's actually animal studies that have indicated that the effects of air pollution may be partially mediated by cortisol and stress activation. And there have been studies even in a human experimental condition where air pollution was shown to causally increase cortisol in individuals who are kind of exposed in a controlled setting. And also in observational studies, they've seen increases in cortisol. But as I mentioned, it's a bit tricky to measure cortisol in general. So I wanted to think, okay, well, is air pollution or specifically fine particulate matter associated with increased levels of allostatic load? And when I looked at the literature, there were 2 studies, one in adults that did see an association with increased allostatic load and one in type 1 diabetic children. And so I think there's a need to kind of explore this a little bit more. So I had an awesome master's student, Selene Muelos, who did her thesis project she wanted to use counterfactual models to understand what the effect of hypothetically reducing the exposure to PM2.5 would be on allostatic load. Using the same study population, it's a little bit bigger because we only screened 2/3 of the population with ACEs because we had an arm to think about what the effect of screening was. But in this population, we could leverage the whole study. We limited the population to kids above 1 because we didn't want to have children who were in utero during the time of the residential linking to PM2.5 estimates. And then we also use a slightly different allostatic load composite, very similar. It's just that she has worked out a pipeline to kind of deal with some of the missingness. And so it's still capturing the same domain. So we just lost one of the biomarkers. But again, here she used targeted maximum likelihood estimation with superlearner that, if you're interested, was a collaboration with a faculty member at UC Berkeley, Alejandro Schuler, who has kind of worked on some of these methods for continuous outcomes and thinking about hypothetical shift interventions that could emerge. So I'll do my best, given that I am a lab person, and so this is my interpretation of these results. So at the top here, if you can see, it's the observed distribution of the annual estimate of the residential PM 2.5 levels. And what is interesting is that at the time of the study, it was conducted between 2017 and 2018. The standards were around 12 micrograms per meter cubed. And so if you look at the mean, it is roughly around the levels of the max at the time, with some individuals being above that level. So what we decided was that because of the revised max to 9 to 10 micrograms per meter cubed more recently in 2024, we wanted to kind of think about what would be the estimated change in allostatic load if you dropped it down to 3, with the assumption that by shifting the max, you would shift the mean. It is a big assumption, but that was our hypothesis. And then what if you did it 1 unit reduction, even more, up to 7 reduction, which would give you 5, which is the WHO levels. What would that do to allostatic load? And so what we see is that with the we had an average of 1.47 in this study of the allostatic load scores. And so with a reduction of 3, we're seeing some reduction, but it's not statistically significant. If we reduce it down to 4, then we start seeing a statistically significant reduction. And as well as with 5, with 6 and 7, we're seeing quite a bit of a wide the confidence interval, so the estimate is less precise. So what our takeaway from this analysis is that this 3 microgram per meter cubed reduction to 9 is not sufficient. If we were to use this measure as an outcome, that would be like a subclinical measure. And so we would say maybe we should be considering a more stringent NAC to at least 8. At least based on this, and I believe that was actually a conversation that the CSAC was having, whether it should be 8 or lower. So all this to say, this is the first study to even think about the association between PM 2.5 and allostatic load among healthy children, at least as this composite measure. And that we would think that we may want to consider potentially stringent NACs, more stringent NACs, so at least maybe 8 or lower. I would like to see more studies looking at this subclinical measure to see if it's something that we could use in other kind of analyses, to think about more subclinical effects that could be monitored when we change or have intervention as well. And again, we're following longitudinally, and so we would want to look at, is this actually changing, given that we are following the kids after there was an actual change, to see if that even models what we would expect. So those were 2 studies in which we were looking at independent effect of psychosocial stressors, and then also looking at independent effects of air pollution on allostatic load. But I wanted to think about, okay, well, does psychosocial stress modify the effects of air pollution on stress biology? And so I wanted to use something that has a bit more literature behind showing a relationship between air pollution and some biomarker of stress. And one of those is telomere length. Telomeres, if you don't know, are the caps at the end of your chromosomes. So think of it as a shoestring. You have your telomeres at the end. And with each cellular replication, they shorten until eventually they get too short and their cells senesce. And so that's why we use it as a biomarker of aging. There's been evidence that definitely that PM2.5 shortens telomere length in children. And there's some evidence that in general that stress does shorten telomeres, but that specifically ACEs have also been related to shorter telomeres. So I had a very ambitious master's student, Austin Lay, who was interested in exploring this research question within the PEARLS cohort as well, to see if early life adversity would modify the association between fine particulate matter and, if childhood adversity would modify the relationship of PM2.5 on telomere length. And to get to the point, since you're already familiar with the study that we were using, in this analysis, we were able to look at different variables or different exposures here of 1 month prior PM2.5 versus 12 months to see if they were like subchronic versus more chronic effects. And we see a relationship that's expected with PM2.5 and telomere length for greater PM2.5 exposure linked to resident phosphorus was associated with shorter telomeres. And we didn't see any independent effects of these adversity measures. And also I wanted to point out that there's this new item or new thing of the domain scores that was created after the original paper I presented was these represent a confirmatory factor analysis of the PEARLS items to see which ones are more correlated with each other. And so we have 3 domain scores: it's maltreatment, household challenges, and social context, with maltreatment being so household challenges being more at the family level, and the social context still having some. It has neighborhood violence, and you'll see why I'm harping on that. They are independent of each other. And so the question is, can you use that information to intervene by understanding which things are pretty much correlated together? And so when he looked at the relationship between PM2.5 and telomere length and whether it was modified, if any of these social adversities modified that relationship, what he found was that the social context domain. So what that means is if you reported, it was ranging from 0 to 4, whether or not you reported any of these 4 variables, so neighborhood violence, housing stability, food insecurity, or discrimination, that modified the relationship with PM2.5 such that when you had reported none of those, the relationship between PM2.5 and telomere was not statistically significant. But as you increase the number of reported social context items, that relationship got stronger, suggesting some modification potentially by reporting the social context, having one of these variables. I wanted to think more about these neighborhood variables and didn't see anything with childhood opportunity index, which would be more of an objective measure. Obviously that social context domain captures more than just neighborhood, but thinking about this more broadly, is there a difference between perception versus objective measures? I think that's something we still need to think more about. So in sum, a lot of that demonstrated that we saw a association between PM2.5 and shorter telomere lengths. Early life adversity may influence the susceptibility of the air pollution effects on telomere length. We didn't see modification by neighborhood opportunity index. And in general, we think that because they're often coexisting, adversity and air pollution within the same community, we need to start thinking about how do you promote healthy aging in these populations overall. But these are all individual biomarkers one at a time. And I think even as a field, our stress biomarkers are inadequate, I would say. And so we're always trying to, we've been studying that since 1970 and still trying to find new tools. And so thinking about, can we leverage exposomics to kind of think more about where this intersection of cumulative risk may happen. We don't have results right now, but I just want to demonstrate we're leveraging a population from Richmond, California. For those of you who don't know, it has a long history of one from World War II. There were ship workers who moved there to kind of help with the war, but they were put in like, unstable housing that ended up being permanent. All of these to say there were a lot of discriminatory practices. There's also the Chevron refinery there, a lot of railways, so a lot of pollutant exposures that coexist. And there's also been a shift in immigration there as well as time has changed. So now it is, there's been an influx of immigration that's happened in the community. And what you'll see here is the asthma ED visits for the different cities, major cities in the same county. And Richmond is here, and it has higher levels across all the other cities in the same county. And as for census tracts that have the refinery essentially in them, that asthma ED visit is the highest. So thinking about all of these co-exposures and cumulative impacts is, I think, critical, especially in Richmond, which is also is an AB 617 community, and rightfully so. We need to think about how to protect human health in this population. I think I'm going to skip to the point of what we're doing with this study, in that we're building a cohort through the exposomics profiling and asthma disease to unveil determinants of disease in asthma, or the expand asthma study. This is one of the disease study sites that's part of the multi-omics health and disease consortium. We're one of the two pediatric sites. The other one is led by Lila Chatsia and Maxong here. And so our site is doing asthma, and we're trying to collaborate to leverage power between the two studies to think about even cross, site comparisons or differences, to think about what are the social environmental contributors to asthma, and can you use omic approaches to really understand that. So this is only a snapshot of everything that's being done, but we're recruiting kids between the ages of 8 and 16 at our site, 200 asthmatics, 100 controls. And so we're really focused on these psychosocial stressors that have been harmonized across the consortium, but some have been specific for our site as well as a number of measures of air pollution. But through the consortium, we're also collecting biospecimens, including blood, urine, and our site is really interested in nasal swabs because asthma biology is most relevant when more captured best through like nasal swabs rather than blood. So we're trying to leverage those biospecimens that have been collected and along with all the rich omic and clinical measures that are coming out of the study, including genomics, transcriptomics, epigenomics, proteomics, metabolomics, as well as exposomics, and a bunch of clinical labs to really try and think about what is the also mechanism that may be underlying some of these independent and joint effects of air pollution and psychosocial stressors. I think the unique thing as I wrap up is thinking about the community engagement piece. We meet people where they are. So our site is actually difficult for the consortium because we are located in the community in a community clinic, and we do not want participants to have to come to us at an academic setting. So we work around them, and so we've been working through that infrastructure we established back when I was in graduate school. So it's a real honor. But also through that pilot study, we learned that we can't just collect data, we have to provide resources and also support. So we've established a youth council and youth programming that I'll talk about in the next slide. Because when we did our first pilot, it was during the COVID pandemic, and the study participants were like, yeah, your study's great, but we really need food and we really need things for our kids to do over the summer. So we've been kind of working with the community to provide that. And that's where I think there's really power in this consortium, because efforts led by Dr. Ong and Dr. Tacker have really pushed this building of a cross consortium community partner panel, which really has been implemented from the beginning till even when we think about what issues we have in the study, plenty of them. And then also thinking about long-term, how do we report this back? What does this interpretation mean to build trust with the community? And I always like to think of the positives. So yes, stress and chemical interactions can be, thinking about, well, what is all the negatives impacts on my health? But we've kind of leveraged our ties with the community to build out a youth participatory action research team, in which there's usually 4 to 6 youth from Richmond every summer that for 8 weeks, they build out their own research program, research question, and we help support them through beginning to end to think about how do they present to their stakeholders, community stakeholders. And so as you've noticed, there's a transition over time to a real interest in greening initiatives. And so that's really where we've been supporting their efforts of thinking about how do they kind of advocate for, the planting of trees within their community. And this summer, coupling it with the greening initiatives, we're working with them to understand where they would like monitoring for black carbon, and so that they can actually present to their the SERP steering committee to think about where we should start even monitoring for this kind of emission source. But with that, I'm going to end and just share, all the amazing people I get to work with, and I'm missing the mode, but all the funding sources that kind of help to support it. That I'll be happy to take questions.
Then you can see if there's questions on.
Yes. Hi, yes.
So did you, you have, your UO1 is focused on asthma. Presumably you had asthma as an outcome, or asthma severity as an outcome in your earlier studies.
Yeah.
You looked at it and didn't see any associations, or?
Yeah, so it's 40% of the population in the first one. That's also because the primary care clinic also has somewhat of an asthma clinic. So Pearls was associated with asthma at baseline. I didn't see a relationship with allostatic load and asthma. And I'm seeing interesting things with the telomere length where it's been reported before where it's a little longer. And that might be because of infiltration of immune cells, but we still haven't figured all that out. Yeah. But I think figuring out, I think also because we have to think about the age of the kids, they're like 1 to 11, and technically you're not really able to diagnose asthma till 5. So that's why it may be, it's a tricky thing to look at in this population. What has been interesting is with another biomarker I haven't showed you, eczema is earlier in the atopic march. Atopic march is that you have eczema, and then severe eczema then proceeds to, develop potentially into asthma and allergic rhinitis. So we're seeing another biomarker. This mitochondrial biomarker is related both to ACEs and also to eczema in kids. And that same biomarker has been associated in adults in the same direction with asthma. So I'm still trying to figure out which of the biomarkers that in my repository might be best for, asthma outcomes, but I think it is promising. And also we have to remember that obesity is a big risk factor for asthma. And so maybe we're picking up something that's a little bit more sensitive earlier on.
Okay, can you hear me?
Yeah.
Thank you for this really excellent talk. I mean, first, like 2 worlds of mine, stand load and exposomics. So my question that I don't have an answer to, but I love your thoughts. Are you thinking of how exposomics can help us, let's say, discover maybe or validate new kind of algorithms to really get at allostatic load? I know that's a tough question, but what are your thoughts on it?
Yeah, I've been playing around with this, one poor PhD student, because we had this pilot that happened prior to, the consortium. So we have it in 70 kids where we have more robust allostatic load measures. They're also a little older, so I'm a little bit more confident in them being more similar to adult measures. So we have we have created an allostatic load score, and in the same population, we receive funding through a P30 grant out of UCSF to run on targeted metabolomics. And so one idea is then, can we look at, you can get both out of the same thing, right? So we did a suspect screen, so we know chemicals. And then also, we still are analyzing that. That's why I didn't present on it yet. But then we can also leverage the biological profiles potentially to then, obviously, I would love expertise on that piece, but you could think about which metabolic profiles are altered through the allostatic load measures, or if you have all these other omics, which I hope the consortium can do, we can then leverage thinking about like, we know potentially allostatic load in its crude form is associated with some outcome, but can we evolve like new biomarkers that may be a little bit better at doing it? So yeah, that's the goal, but you know, thank you for your question.
So I have a question for you. Especially around the ACEs, you know, there's a couple of different domains that you're touching on there. Has there work been done either in your group or more broadly, trying to couple qualitative methods with it to really dive in deeper with the participants of how these experiences might have affected them? And you know, these questions around like, you know, timing, you know, resiliency that was built up maybe after the experience, additional ACEs. Can you comment on that a little bit?
Yeah, I think you're getting at one, we know, yes, there's a need for the qualitative aspects of it. I don't think this study is doing that. We do have the family board, and so we get their like, you know, their lived experiences. We haven't coupled it, you know, with the biology also. And I think I'm not answering your question exactly the way you want it, but what I'm thinking is also, we know there's so much heterogeneity in the response to even just one adversity measure. So like that screener alone is not, we know it's actually, there was a study in JAMA Peds that like that screener alone is not predictive at the individual level. So we really need to start thinking about what are these contextual factors, right? So do we ask, you know, why, like, what are these sources of heterogeneity? So I think we're trying to figure that piece out, but I think you're right. Psychologists tend to do a much better job of like thinking about what are capturing, like, what are some of these sources of variability. But in a primary care setting, we haven't done a great job of that.
Is the consortium thinking about incorporating some elements like that?
Like qualitative
Yeah.
measures into it. We have a lot of questionnaires that are harmonized, but I don't know that we capture it like, I don't know. I would say like in our family so we were funded for the ACEs one through the California Precision Medicine Initiative. And so in that piece, we have a family board to then think about resources that we could provide. So there may be some elements of the experiences that we could, I mean, that people kind of share. But I would have to get back to you. I'm laughing because I was just working with a psychologist on this like space, and I'm sure she has like the right answer for you right now. But I think it's important to do that coupled work, because these screeners alone are not, they're imperfect. We know it. It's just, even just getting them implemented in a medical setting has been a challenge. Yeah, I welcome. Yeah, I appreciate it. I think there was another hand. Yeah, just
Yeah, so can you talk a little bit more about the telomere length and immune infiltration and how that might also be impacting single cell in the blood samples? Is that something that you?
Yeah, so we could theoretically look at this a little bit because we have blood cell counts. I haven't dug into it a little bit, but the hypothesis here is that if there's a lot of inflammation that's going on theoretically, and these are things we haven't explored, it's just, you know, hypothesized why you might, because if you read the telomere literature, sometimes things bounce around, like some things get longer. That makes sense in the case of cancer where there's more localization, but in terms of like normal homeostasis, why that might happen is because you have newer cells that have come in, and it could be because there was so much so much inflammation, it caused senescence of some cells, and then the hypothesis that new immune cells are coming into the system. But yeah, you need to like trace the cell types coming in, and we haven't done that, but it is a hypothesis that is theorized there. Now, also to caution, it was in buccal cells, but that still doesn't mean, it doesn't, they're buccal swabs, but that doesn't mean that you don't have immune cells coming in. So that those are questions that could be further explored, yeah. There's no question. Okay.
Any additional questions?
Maybe just one other, just maybe more of a comment or rhetorical question. So you started out your presentation talking about high rates of asthma in the Bronx. And then you showed the Cross Bronx Expressway. And so, you know, we always think about hazard ratios or relative risk. We don't think a lot about attributable proportion. You know, like that's, I think that's sort of the question that you posed to begin with. I don't know, it's just an idea, you know, in terms of what, you know, what we focus on.
Yeah.
And what we really like to, you know, that sort of global heterogeneity, you know, how much asthma can we actually explain by looking at these interactions? I don't know if you have any thoughts about that.
Yeah.
There's no question really in it.
No, it's a good one. Yeah, I think, yeah, the question being, what is driving essentially these, again, I'm my epi, I'm trying to get at this a bit. I think it is an important thing of trying to figure out what is contributing to, you know, the prevalence that we see within the population. And so, figuring out, because that's the problem of cumulative, right? And so just trying to figure out what is like contributing overall, I think would be helpful, yeah.
Exactly, kind of around the same line, and I think the one study that you showed that was the TMLE approach, the decomputation approach, or the counterfactual approach, kind of got off this, like, what happens if you were to reduce air pollution exposure to some hypothetical standard? What happens to your count in the your outcome, your distribution of your outcome? A lot of what I heard is like, you know, we're trying to look at it this way, you know, we have imperfect measures, both on the survey instruments and also our biological measures to try to get at these themes, and especially more broadly thinking about the exposome and the totality of exposure. How do you see the field shifting, especially in, you know, the 10+ years of your career so far in research, of will we be able to get to a point where we can account for, you know, a variety of different factors to, if we can give individuals the tools to be more resilient to adversities experience in their lives, or better remove, you know, the adversities or remove air pollution. Do you think we'll get to a point where we'll be able to analyze and estimate with some level of accuracy what would happen under these different kinds of scenarios if we were to do these interventions?
Yeah, I mean, we're in its infancy, but I think we'll see with more time, right? If we're able to kind of, I think that's where we would be able to then estimate, you know, what would be. I think, yeah, I do think that that's where like more measurement essentially through exposomics or potentially could capture more measurements, and then we could then start understanding like what are, you know, the proportion that might be explained by X unknown compounds, right, or whatnot, or these things maybe are not as much of a focus. And then should we then think about it. We've done it also for like green space and whatnot, and like how much of that would be reduced, in a hypothetical scenario. And so like, I think all of understanding more of what contributes to the disease would then allow us to understand what proportion would be explained away if we were to kind of remove it in the population, I would imagine.
Do you feel like the statistical methods have kept up with the biological methodology to try to be able to disentangle these things because they are so?
Well, that's the bottleneck, right? I think where I think colleagues here, I mean, I've seen some of the work that, like Lucid, and the work that you're all like doing here is trying to really understand how to kind of analyze it to kind of get at some of those questions, right? Of like, can we then leverage that understanding? Because right now it's so high dimensional, right? It's one of the problems. And yeah, we're getting a lot of data, but like, how do you analyze that? How do you figure out what's driving it? Yeah. I think we're still working on that. Hopefully we can figure something out with more data, but will we then end up with a problem of like, you have so much data that it's hard to interpret? Yeah.
Does the consortium have an arm around like the statistical analysis?
Yeah, there's the data analysis core, so that's part of it, like implementing cross multi-omic analyses. So we'll see what they come up with.
Any last questions? And we think we've heard from a lot of faculty, but there's any trainees who had a question perhaps? Okay, I'm not seeing any and I won't force you to stay. We have lunch after this and I think the lunch boxes are outside. And so for the next half hour until about 1:30, you will have an opportunity to have lunch with Dr. De La Rosa. And then afterwards in the afternoon, we actually have a couple of focus meetings. If you're open to anyone, if you're interested in these, it's going to be upstairs in room 222. So just the corner upstairs conference room. At 1:30, it's allostatic load and biomarkers of stress. At 2:30, it's more broadly biological pathways. And then at 3:30, on the multi-omics for health and disease consortium, we'll be referencing during today's talk. And then that'll be it. So thank you, everybody.
Thank you.
So, it is not as a biological concept.
The calendar event or the person who suggested one?
Yeah.
Which one? Sorry.
Oh,
The person who suggested the title?
Yeah.
Here.
okay. I was gonna say, because if it was Lita, then it's kind of duplicated.
Oh, the other one? No.
But-
It was, we were trying to get at, and give you some other.
I know. We're gonna just keep talking on finally.
So a lot of people were. Allostatic load, right, we put that one as the one right after. And then we made it more broadly biological pathways. Fit in the co-cortical. And the other things that I did not find out. And so that's why I kind of delved into that.
And Carrie's here?
Carrie is actually, I think she's on vacation. She thought she was gonna be here, but I think. But she said she was probably good. No, I don't.
You?
That's what her.
Okay.
Hopefully we'll be able to attend.
And
And
probably
really,
the headphones. Or not.
I know like a suggested title, so the conversation we had is just the opportunity to have some fun. So, yeah, are you gonna be into the afternoon meetings?
Yeah. I'm planning on this one. This one, I don't know if I can.
So I. Yeah.
It's a little. And then you guys are gonna go to dinner about 5:30.
Whenever.
Go after.
Yeah.
Lately, we've been going right after the meeting. It's awkward to have the hour block.